Greeting to all. Welcome to our channel, Mountain Heights. Don't forget to subscribe onto our YouTube channel. My name is Teacher Bernard, and our topic is protein synthesis. Now, this process of protein synthesis involves the DNA, the RNA, and the genes. It occurs in mainly two main steps. That is translocation and transcription. Now, remember, the proteins are the ones that determine our body activities. They form what we call the enzymes, the hormones, and other organic components of the body that control every feature of an organism. Now, the genes, how does the genes influence our activities? The genes influence the proteins to be formed, and the proteins influence our activities in the body. Now, this process of protein, we're going to see how the DNA, the RNA, and the genes form the protein. I said it occurs in mainly two steps. That is, transcription is the first step. It's called transcription. The first step is called transcription. Now, this process occurs in the nucleus. Now, in this process, we are trying to form what we call the messenger RNA. Is our aim of this process is to form what we call the messenger RNA. Now, this, from the word messenger, it carries the gene codes from the DNA that are going to describe a protein to be formed. Now, how does this process occur? It begins by unwinding. Remember, our DNA is a double-stranded molecule. The DNA is a double-stranded molecule. DNA is a double molecule of a helix structure. Now, it begins by unwinding. These bonds that are joining the strands are broken to form free strands. Now, the enzyme is called helicase. It's our enzyme that breaks down these bonds. The enzyme is called helicase. It's called helicase. This enzyme comes and breaks the hydrogen bonds to expose these strands. Remember, here we have our bases. For example, we have adenine, we have guanine, cytosine, and thymine. We copy again our adenine. And here we have the thymine, we have adenine, we have guanine, cytosine, and thymine. Don't forget to mark this combination. Thymine goes with adenine and guanine goes with cytosine. And the bond here, they are triple bonds. You have double bonds. Now, I said this enzyme comes and it breaks these bonds, exposing the bases freely. Now, free bases come in and pair up. These bases occur naturally in the nucleus. Now, they will come here, shall have a new base to come. That will be guanine. Have our, remember, now here we have our adenine. Here we have our uracil. Now, since we are forming an RNA, adenine will go with uracil, not thymine. Thymine goes with adenine if we are in the DNA. And if we are forming an RNA, adenine goes with uracil. This is what will be formed. You see, a new strand is being formed. The enzyme comes, breaks the DNA, exposing the bases freely. Now, free bases come and they begin to pair up with this opened bases. Now, you have a protein called polymerase. It's called polymerase. This is the enzyme that that helps to form this base pairing of the newly formed RNA. Now, after forming this RNA, it now leaves. Now comes and leaves the DNA. Now it gives us the messenger RNA. Now the DNA winds again and form and go back to its helix structure. Now this messenger RNA contains the gene codes that are responsible for the protein to be formed. Now all this occur in the nucleus. Now afterwards, this messenger RNA leaves the nucleus. It leaves the nucleus. We said this process occurs in the nucleus and we form what we call the messenger RNA. Now, before this RNA leaves the DNA, it is first edited to remove the intron and the exon, of which we talked about in the last lesson. Now, after this RNA leaving leaving the leaving the nucleus, it it becomes attached to what we call the transfer RNA. It's attached to the transfer RNA. This transfer RNA, its function is to transport this RNA from the nucleus to the ribosomes. Why? Because this RNA can be combined with other bases in the cytoplasm. Therefore, it is attached to the transfer RNA. And this transfer RNA has what we call the anticodons. These anticodons are complementary to the codons of the messenger RNA. Now, it becomes attached here and then it is moved to what we call the ribosomes. And there, you have what we call translocation. Having translocation. The word translocation means we are going to read this messenger RNA to translate it into the amino acids that form the protein. Now, this occurs in the ribosome. Remember, ribosome has two subunits. We have the small one and the large one. The large and the big subunit. Now, this messenger RNA, comes. Remember here, you have our bases. You have guanine, adenine, and uracil, and many others. This is our messenger RNA. This is the transfer RNA, and this is attached in the ribosomes. Now, this one becomes attached to the small subunit of the ribosome, exposing these bases to the large unit of the ribosomes. Now, we have what we call the ribosomal RNA. How what we call the ribosomal RNA. Now, this one comes and reads these codes and translate them into the amino acid. It comes and translates these codes of the messenger RNA into the corresponding amino acids. Now, these amino acids come and they begin to form just below these bases. These amino acids come and begin to form just below these bases. Remember, this is a longer chain. Others come and begin to form amino acids. Now, these amino acids come to begin to pair up, forming what we call the peptide chains. Later form what we call the protein molecules. Don't forget to subscribe on our channel called Mountain Right? The question answer services.